All right, hello everyone. Welcome to part 2 of STAT's 3-part digital event series, Combating COVID-19, sponsored by Pharma. Thanks for being here. I'm Matt Herper, and in just a minute, we'll be joined by Pfizer's Michael Dolson to talk about the wide range of approaches Pfizer is taking in the fight against COVID-19. But first, a few quick housekeeping items. At the bottom of your screen, you should see a few icons. There's a chat icon and a Q&A icon. Use the chat feature to chat amongst yourselves or to send a message directly to the STAT team. If you're having any technical difficulties, please let us know in the chat. But do not put your questions in the chat. If you do have a question that you would like to ask me or Michael, and I hope you do, please use the Q&A feature to send that question to us, and we will try to answer it live. Again, we will not be taking questions from the chat, so please type your questions into the Q&A. There are already a few great ones there. More are welcome. Thanks so much to everyone for being here. Before we get started, I'd like to first welcome Richard Micciche, Chief Medical Officer and Executive Vice President of Science and Regulatory Advocacy of Pharma, to introduce today's session. Take it away, Richard.
Thank you, Matt, for that introduction and for hosting today's webinar. Today, as we wrestle with the challenge that's in front of us, I am optimistic on how the biopharmaceutical industry and its science will provide the answers we need to beat COVID-19. We need as many shots on goal as possible, but it's the identification and development of a coronavirus vaccine for patients which we know will have the most transformational impact. The road ahead is challenging, with only a 5 to 10% success rate, but already we have more than 400 global clinical trials for COVID-19 treatments and vaccines, and over 200 of these trials are testing pharma member company products. At least 6 vaccines for COVID-19 have entered human clinical trials, and many are planning to begin human trials this year. This rapid pace of accelerating vaccine candidates is unprecedented and showcases the biopharmaceutical industry's unique capability to act swiftly in this pandemic. In the midst of this work, our companies are also steaming ahead on therapeutics, which are complementary to vaccines to keeping people safe and healthy. A number of different antiviral approaches specifically are being taken to reduce and reverse the effects of the virus in infected people. These include antivirals that have previously shown promise as treatments for other viruses, including coronaviruses like SARS and MERS. And we should start seeing the results of the first of these well-designed trials as soon as the next few weeks. Companies are in a position to move quickly because of their vast experience working on such similar viruses, which has dramatically improved our likelihood of success. And our industry has invested billions in technologies that have significantly shortened the time it takes from identifying decoding viruses to developing a potential vaccine to be tested. All of this is happening through extraordinary collaboration across our industry and with U.S. and global health authorities, including the NIH, CDC, FDA, and others. We have a unique R&D ecosystem in the U.S. that fosters robust collaboration between industry and the government, and it is working well. Of course, our industry is doing more than research and development for vaccines and treatments. We are also working with other life science companies to develop new diagnostics, both to detect the virus and to detect antibodies to the virus. We are expanding our unique manufacturing capabilities and ramping up production capacity at risk. We are working with governments and insurers to make sure that these potential treatments and vaccines will be both affordable and accessible to patients. Every one of our member companies is taking steps to help address this crisis. If you are interested in learning more, we keep a full list at pharma.org/coronavirus. And now I'll send it back to you, Matt. Thank you.
Thank you, Richard. I'd like to introduce our featured speaker for today's event, Michael Dolston, Chief Scientific Officer at Pfizer. Thanks for joining me, Michael, and a reminder to all of you to keep submitting questions through the Q&A feature at the bottom of your screen. And I am going to start asking some of those questions. There are a bunch of great ones, Michael. Now, several of them stated most succinctly by Kathleen Dorn here. What do you think of Gilead's data? A little more of a, I'm not sure that you'll want to directly comment on that, but I wanted to ask, given the success that remdesivir has apparently had today, and obviously we don't have any details, what do you think that means? You have a big antiviral effort going on yourself. What do you think that that positive results, any positive results in remdesivir mean for the ongoing effort to create antiviral drugs here?
Yeah, thank you, Matt. First of all, I welcome, and that's the position of the Pfizer company, progress by anyone in the field with the novel therapeutic and vaccines, given the urgency to come with a health solution for patients and society. So my fingers crossed that once we read about the Gilead remdesivir data, that they show some really dramatic effect. And of course, we need to, as you have seen and many of us, that a lot of data is floating around in various formats, often rumor type of data, sometimes small trials that doesn't have a control group. So I think this will be, to the best of my knowledge, the first somewhat more substantial trial by NIH with a control group. So it should allow us to feel more scientific rigor in the data. So fingers crossed for positive data, number one. Number two, the magnitude of data, whether it's incremental or more dramatic. And number three, which endpoint. In the end, what you want to see, it is that your treatment protects patients from progressing to severe disease, intensive care units, and fatal outcome. So that also is very important to look at all the critical endpoints. I think a success here boosts the entire field. At Pfizer, we work on a different mechanism. And we would certainly think that the success here would make all of us believe that we have a good path, and we would be then trying our utmost to move quickly and even augment the clinical benefit we can provide to patients. And then for many patients, of course, we know that it's not just one treatment when you suffer from viral diseases. Often we end up in combination treatment. That's been true for similar virus diseases like HIV and hepatitis C. And I will be happy to comment on our thinking how a drug type like remdesivir could combine with a different drug type that we are working on, Matt.
Mm-hmm. So we'll go ahead. I think that's a good thing to follow. So let's talk a little bit about imagining what a combination therapy here might look like and how quickly that might roll out. And isn't it a bit of a challenge? It's great that we have so many irons in the fire, but there's so many different molecules being tested. How do we decide what to combine with what?
Yeah, I think what you start with is to do in vitro tests on cell lines that are infected with SARS-CoV-2. And most often, what is promising is when you combine two different mechanisms that then disable the virus in two different parts of its machinery to try to take over the human cell. And I think like a drug like remdesivir works, being a nucleotide analog, it works particularly on the ability of the cell and its on the virus and its polymerase to replicate its RNA and create more viral particles. A very important mechanism. We're working on a protease, And what fascinates us is that it's a really unique enzyme for these RNA viruses. And a counterpart doesn't really exist in the human cell. So we hope we could dose high, hit it hard, and not have cross-reactivity with our own cells. And we do have preliminary data that when you combine the drug class of polymerases, to which remdesivir belongs, with a protease, we see additive effects. And that's a very important sign that a drug like our protease inhibitor could be active by itself, whether similar, less, or better than the first, hopefully soon to come, active drug on polymerase. And we should have positive hopes for that when you combine it, you would see viral load drop faster in patients, and more patients would be able to eliminate quick the virus. And Matt, that's so important. The longer the virus stays around and starts to trigger our immune system, the higher the risk is that they progress to this hyperinflammation syndrome that makes a very severe acute respiratory distress syndrome and a fatal outcome.
So, as you might expect, we're getting a lot of questions about your vaccine work. You guys had some good news yesterday and today about progress of getting trials running there. But I think Tony Artino asks really clearly the question I'm hearing mostly from people outside the industry, which is, what is the real potential timeline for a vaccine? And he says none of this happy talk that about 6 months, which he has a hard time believing. What's a conservative answer? How long are we going to have to wait for one? So I mean, maybe start with that big point, and then let's talk a little bit about what Pfizer's doing. And there are some specific questions about mRNA vaccines and about the Oxford work.
Yeah, I think it's a great question here. And I think as a scientist and any communicator in this field, you need to start with realistic but stretched ambition. And you need to challenge yourself to think, how can I do what we did in the past in a new, faster way? And how can I do things completely differently? And I had the same view as the colleague that raised this question, that it would take, I think we heard like Tony Fauci saying like 12 to 18 months. And actually, our CEO, Albert Pola, challenged me and my colleagues and say, look, there is a flu season coming up this fall, maybe October. Take that as a challenge and work backwards. What if both flu and COVID-19 disease would hit us at the same time? And interestingly enough, you heard Robert Redfield bring up that as a major challenge for society and healthcare system. So we took that, and that helped us to identify a path. From now, we started to dose last week in our vaccine trial. So that helped us, in roughly 6 months to make a map how we could, in October, be ready with, if things go well with our clinical data. And I must say we've had tremendous favorable regulatory dialogue from the highest level of leadership there, Commissioner Hahn and Peter Marks, in encouraging us to come with urgency into this vaccine planning. And we actually see a path where you work, and I say it not just as we Pfizer, I say it more as your question what was put to me, how you could do those vaccines. And the key is not to think about that you do clinical trials step by step, but you do them in a continuum, and you adapt the design according to data, and that you are transparent and share when data comes in with regulators to get early dialogues that allow you to quick change the format of the vaccine, the dose, the number of vaccinations, and expand those parts of the trial that looks more promising. So we do certainly believe that by October, through the human study and some supportive studies on surrogate endpoint, that there could be a compelling data set for regulators to review, and that you could either go a path of emergency use or actually accelerated approval, which give wider access. Now, if there is an approval, of course, the question is, can you have enough vaccine available to start treating millions, tens of millions, and 100 millions? And that's another question we can continue with.
Well, that is actually my next question, and I would have said billions if we're talking about on a global level and actually vaccinating everybody against this vaccine. It's a lot of people, right? So what kind of production capacity would there be for, assuming that only one vaccine proved effective, what kind of production capacity would there be in that short time frame? I know you're not building at risk, that you're building out production capacity, but still, that's a lot of people.
Yeah, and clearly for a company that has never done this before, I do admire biotech companies that are throwing themselves into this important battle against SARS-CoV-2. It is, of course, a way of building the airplane while you're flying. But please remember, at Pfizer, although it's a different technology, we have been one of the companies that have supplied the larger part of the global population with pneumococcal vaccines. So we are very used in thinking from the lab, clinical studies, and billions of people that may need and benefit from your vaccine. So, what will happen in October is probably that we will have, pending success and regulatory support, we will have millions of doses. And you would need then to think with advice from public stakeholders, who is most at risk, Matt? Mm-hmm. Who is at the front line, and which regions of the world, which cities in the United States have the highest hit of a potential bounce back of the virus. And later this year, we think we can be at tens of millions, and then you could expand into the next tier. And as we come into 2021, 100 millions, and onwards, of course, billions. Now, there may likely be 2 vaccines or 3 vaccines. So you have to think as a team of vaccine producers. If you're only one, you obviously have a much harder logistics. But I think we'll need to also coordinate. In the US, we have a great federal agency, BARDA, that has as one of its major remits to advise companies and even help to lower at-risk scale-up and procurement of vaccines. And I think we'll see similar initiatives in Europe and the developing world. And we know, groups like the Gates Foundation, CP in the rest of the world are putting a lot of work into that. Mm-hmm. So I see a roadmap that will come together soon, that will hopefully have a plan to go to billions as soon as we can.
So just to explain to people a bit about your vaccine, which you're developing with BioNTech, it's an mRNA vaccine, which is a new technology. It's the same basic idea as the Mrna Vaccine, which is also in trials. There are a few others, and this is it's a really cool idea that you take RNA, which is the messenger between DNA and proteins in our cells, and use that to make proteins in cells. And there's been some early data showing that this can work for a vaccine, but there are no approved vaccines. And you have a somewhat different approach from Moderna, with multiple antigens and also 4 different candidates you're developing. Give a really quick overview of that, and then I have a question from an attendee I want to get to.
Yeah. So Matt, I think you started really well there to describe that we were intrigued by mRNA vaccines because it's fast to synthesize them. They're completely synthetic. You don't need a cell to produce them like proteins vaccines, or when you infect and use inactivated viral particle, etc. So that means that this technology can adjust quickly its component, pending if SARS-CoV-2 would change sequence. And I think we need to be prepared this is just not going to be a couple of battles and it's over. There is high risk that this virus is going to be with us as we learn to inhabit HIV as a lifelong threat or something we need to manage. As we got used to see the various flu viruses come and go season by season. So we wanted to invest in something that have the ability for the future to be a long-term, very fast-reacting defense against the coronaviruses. Now, you're absolutely right. There is a less proven success with this new cutting-edge platform, but we have been working 2 years with BioNTech on it for flu. So we felt we had a good understanding in solving the most apparent bottlenecks. Mm-hmm. And then I think as we started to see the SARS-CoV-2 as a major challenge, our next piece was that we thought this platform could so uniquely be positioned for this pandemic approach. Mm-hmm. And while Moderna, that you mentioned, a great company, has chosen just one vaccine format, we learned that in our studies that it's hard to predict for a new disease that's 4 months old and for a platform that is kind of the next cool thing in vaccine platforms, exactly how to refine it. So we went to the clinic with 4 variants where we have chosen variation in the RNA itself, in what it codes as an antigen, while we have kept the lipid formulation fixed because we did a lot of work on that over the next years. So that's why I feel enthusiastic. We can cherry pick maybe in 2 months the 1 or 2 most promising of these formats and then move that forward. So that's why we made this bet. But as you know, R&D is a risky business, and we got intrigued that this is a technology for this type of challenge.
So I usually don't allow anonymous questions, and I'm trusting this anonymous person that they're getting the citation right to Merck's call from yesterday, because I wasn't on it. But they say that Roger Perlmutter, who I know we both know well, said that he worries that mRNA vaccines might not have enough persistence or immunogenicity. And I thought throwing a question like that from Roger, who we know is familiar with all the platforms, he has your job at Merck, was worth doing. So do you have any answer to that on the?
Yeah. I would probably have had that same view maybe 1 to 2 years ago as Roger, but we were curious to embark in this space because of the tremendous potential it has. And I think we've gotten increasingly confident that this will not be limitations in the platform. And the 3 different RNA formats includes RNA that is highly immunogenic. It includes RNA that's moderately immunogenic that allow likely the cell to stay as a vaccine factory for a longer time, while the first one will elicit more of a dramatic vaccination, which could lead to a strong initial response, but less sustained. And interestingly enough, the last one is what we call self-amplifying RNA. Beyond coding for the antigens to raise an immune response to SARS-CoV-2, it also encodes for an enzyme that allow the RNA to multiply. So we think it may be, once it's refined, hopefully in these studies, be able to expand 50-fold. You may need 50-fold less with this self-amplifying than with a traditional RNA. And we do think from preclinical studies that this can be a type of vaccine where you even should aspire for a single injection with quite sustained activity over time. So, this is what we have learned. Nobody can predict exactly how things plays out for any platform, but these were things that were on my mind, but will not in the long term be a bottleneck for this platform to the best of my thinking, and hopefully not short term.
Great question from Thomas Seo here. Isn't the functional question of having a vaccine, whether folks would be willing to take it in an illness where the vast majority of people have milder symptoms and many are asymptomatic. Doesn't a vaccine have to be really, really safe, and doesn't proving that take time? Even if you can prove efficacy in the next 6 months, don't we need a pretty long and large safety study before people who are at low risk are going to be willing to take this vaccine?
Yeah, you have two important components there, or there's actually three in it: safety, and the other is how it protects those that are at risk for disease or get exposed. And number three, where do you feel solidarity with the billions of people on this planet to not be locked in like you, I, and Rich was previously at home? And let me just briefly touch on each of them. In October, we'll likely have thousands of people in safety as a good start. Mm-hmm. And I think as you roll this out, you won't be able to vaccinate everyone at the same time. So whether it's approved, accelerated, or emergency use, you will quickly learn a lot by comparing regions that have access to full vaccination from the start and regions that got the vaccine maybe with a slight delay. So you will pick up a lot of learning about what unexpected but likely more rare adverse events, because we would hope to already by then have thousands of people vaccinated. So you are looking for more unusual events or things that could happen that you didn't expect when people acquire various type of illnesses such as COVID-19. The other part is, of course, it is in great interest for those many that are risk groups to avoid ending up in the hospitals. Not just, even if we have great medicines by then, not all will be cured. And it's a dangerous environment to be in the hospital with all other bacterial and viral strains circulating. Mm-hmm. So you would probably like to start with frontline public health servants, patients that, or people that are above 60 years, people that have impaired immune system, chronic obstructive pulmonary disease, obesity, hypertension, and soon you're probably talking about 30–40% of the population. And finally, the rest, if we don't make a vaccine for a disease like this to a common procedure that we all feel loyal and have solidarity to do, what will happen is that the virus will continue to circulate constantly in the population, and you will every year have new people that are hit. And by time, those that felt they were not vulnerable may have gotten diagnosed with hypertension, and suddenly they are vulnerable. So I do think that this would be something that we would be, to the great majority, able to take on as a joint objective. And I will just end on looking at why would it be something we all would feel we should buy into. Well, look at today, I think we are at 58,000 dead Americans. It's so sad. In 4 months, Matt. Mm-hmm. And it's not just the biggest pandemic since 1918 or so. It's also the same number of individuals in US that got killed in 10 years in Vietnam. It's more individuals than 3 years in the Korea War. Mm-hmm. I'm just using the American numbers. It's with the same empathy and sadness, I will say, about the tens of thousands that succumbed to the disease in Italy, in Spain, in China. I use it just for an example to, as you raised here, will patients buy into it? Will we be able to communicate it in US? So I do think so many will be so engaged. Everyone will know someone that suffered.
So another question on that I think is is there, from Mike Boss, is there any concern about recombination between the vaccine and the RNA of the virus itself or other coronaviruses? I'd like to expand that a little bit to general worries about safety of vaccine, and also something that Tony Fauci has brought up, which is that in rare cases, vaccines can make infection worse. We saw that with dengue. How do we make sure that that's not happening, that there isn't some untoward effect from the vaccine that's worse than the disease as it is now?
You know, that's it's really a good question, Matt. And let's start with the last one. You know, we have been incorporating that into our selection of the 4 vaccine formats. Mm-hmm. And two of our 4 vaccine formats contains a very small piece of the virus, and the piece that allow the virus to attach to our cells before it can internalize. The virus doesn't have a life on its own. It needs to enter our cells, and then unfortunately it takes over steering of our cellular machinery. Mm. So this is called the receptor binding domain, and it's the tip of a piece, a spike as it's called, that it sits on the envelope of the vaccine, and almost like a knob tries to find a binding receptor on our cells. So our data showed that if you take this smaller fragment, you have multiple places where antibodies can bind, but almost all of the antibodies are neutralizing and paralyzes the virus. I think there is a lot of belief when you have antibodies that binds on the virus, on regions that are not involved in a function, then you may, unfortunately for some vaccines, enable the virus to enter through natural receptors for antibodies. Mm-hmm. So we decided to go with the entire spike protein that's larger, and a piece that is about a fifth of that, 20%, and compare and be extremely careful initially and monitor. Our data so far have shown promise that this is unlikely to be a challenge for this type of vaccines. And there is one more component. We have learned that different vaccines may tip the immune system into different directions. And if you tip the immune system into a direction that's more typical for what you see during allergic reactions, you're more at a higher risk of having those type of vaccine-induced enhancement. And when we monitored in preclinical studies, the mRNA system tipped it into the opposite direction, mm-hmm. being more antiviral than risk of amplifying. So of course, fingers crossed, we'll be measuring in humans, but this again was part of why we really liked this mRNA approach, why we wanted to select a few different formats to minimize the risk of those things, to limit our path forward. And I think that's really unique for us, this comprehensive approach.
Just because I always get these, we always hear these kinds of questions, I think it might actually be worth addressing this from Luna. A prevailing rumor, which this person doesn't believe, that vaccines are really an attempt to chip and follow people. Can we say definitively that you're not trying to put microchips in people? I mean, we're laughing, but people really are. We know how big a problem vaccine hesitancy is.
Yeah. You know, I think vaccine hesitancy is it's sad, it's cruel, because often it's driven by adults, and those that suffer are either not well educated, or it's young kids that think what they hear from an older, mature person. Vaccine has been shown to be, you know, the greatest public health treatment, and save millions and millions of individuals with a rather simple intervention. And there is certainly no techniques available that we know about, or anyone would consider to use them for the single purpose to protect people, protect you and me, protect neighbors from devastating infectious diseases. And in this COVID-19, to restore some kind of normal life back into society. I just welcome those that for some reason are part of this vaccine denial community to take some time and speak to people that are scientists, not necessarily to vaccine scientists, if you don't trust those, but start the discussion by not being biased, not bring any of your ideas of the past, because that's the biggest threat, I think, in society and in fact-based decisions, that you have a bias from the past. And we have actually done a lot in our company to try and train people to not have biases, so you don't assume things, but rather look at everything with fresh eyes.
Somewhat related, but more, what about safety for pregnant women and babies? How much of that will be available in the early stages of having a new vaccine?
Yeah, you know, we used to be careful about vaccines and pregnancy, but we have learned over a number of years now that pregnant women do very well with the more traditional established vaccinations. And we are actually pursuing a number of important vaccines for pregnant women. And I actually mentioned one of them yesterday on the earnings call, respiratory syncytial virus that cause in the United States tens of thousands of hospitalization, some very severe for the newborn. And we are immunizing pregnant women, and we think it's a safe procedure based on recent experiences. So now we need to obviously convince ourselves that there is no component in the new SARS-CoV-2 vaccines that are exceptional, but a general answer would be we think many of those vaccines should be able to be used during pregnancy, and the best is usually the last trimester, because then the antibodies from the mother would pass on to the child, the newborn, and stay there for maybe 6 months and protect it from being infected, and maybe then make the grandparents that were looking forward to the new newborn getting infected and end up maybe in a very difficult hospitalization.
Mm-hmm. So a good question here from Amy Hunter that I'm hearing a lot, and affects both your antiviral and vaccine development. Is there any likelihood that we're looking at different strains of this virus causing different symptoms in patients? I think a lot of people struggle with the really vast difference in outcomes for different people infected here. Could any of this be caused by different viral strains at this point? And does that affect how you develop vaccines and therapeutics?
I think that's one of the really top questions that we spent quite some time on thinking how to prepare for this, and it's likely already happening at a smaller scale. So starting with a vaccine, where we are following the genetic sequence from a number of countries, initially provided by China, from different isolates of SARS-CoV-2. And in the vaccine components that we selected, we have seen some small variations, none of them affecting the regions that we think are most critical for inducing antibodies that can block the virus. We see other parts of the virus starting to change shape. And it could be that those give rise to some different symptoms as it's adapting to the environment. So that's, I think, something that will be very important to understand, and whether that could be that certain regions report different outcomes. And finally, also for the antiviral, where we work on this protease, a real important gateway in the cell, in the virus when entering the cell, whether to be disabled or develop its full potential to spread. we have seen also there very few mutations. So it tells us that we are hitting the virus with a vaccine and a protease drug in an area where it's hard to change. If it changes, it starts to lose its ability to deliver on its objectives, to expand, to infect more cells and to grow its population. So I think what we have carefully chosen is not likely near term to slip through our fingers to something that escapes. But I think we should be prepared for, in a few years, that could happen. And there is even a more scary outcome. The coronaviruses, they are not as old as other RNA viruses like HIV or hepatitis C or flu. But over the last 10,000 years, they have been extremely fast in adapting to different hosts and jump from different species, including to man, and establish a whole community of viruses. And they have now a capability to recombine between them. So that's something that likely will happen every few years. A different one will pop up. And that will require a reshuffling of a vaccine, possibly even of small molecules that are antivirals or antibodies. Hopefully some of them will have enough coverage to help us to keep the initial storm at bay while we retool the platforms that we now are developing. I hope that painted a realistic threat, but also some optimism that we will refine our defense systems and be able to come faster into combating the virus than what happened now, of course.
I mean, something I think is worth emphasizing about that is that some people are surprised when they look back and they see predictions that coronaviruses might be the cause of the next pandemic. That's why they were suspected, because they exist in a lot of animals, they can pass to humans, they mutate quickly. It was always kind of likely that they would be the bad actor, and we've known that another pandemic, be it flu or this, was something that would eventually happen.
Yeah, just a brief comment, because I thought there was such a brilliant thing you mentioned there. You know, the coronavirus as a species tried twice to expand in the human population. Mm-hmm. SARS around 2003, and then 9 years later, MERS. And they were too limiting factor, they were too lethal. So the patients died too quick to favor the goals of the virus to spread. And they were not contagious enough. Mm-hmm. And now the COVID-19 through SARS-CoV-2, through changes, was selected because it seems to be active both in the upper and lower airways. And the upper airways, of course, when you cough, you sneeze, improves its ability to infect other people. And then it was, evolved. So as you said, maybe 20% get severe symptoms and too many of those die. But a lot of people with mild, almost asymptomatic disease. And that's allowed it to be this invisible enemy. So, it learned the third time to really expand beyond just small regions, the species.
Let's talk a little bit about the third approach that Pfizer is pursuing, which is not drugs directly against the virus, but drugs to tamp down the immune system when it over-responds in this condition called ARDS that is part of what really hurts patients in the hospital. We've had mixed data on other medicines taking this approach. There was kind of some disappointing news from Regeneron on their IL-6. There was also a French study for which we haven't seen data yet, saying that maybe with Roche's IL-6, Actemra, that there was efficacy, but there are a lot of, you guys are testing Xeljanz, which is a rheumatoid arthritis pill. There's people are trying BTK inhibitors, which are mostly cancer drugs, but are being tried in other autoimmune diseases. There are a lot of drugs that work on the immune system this way. What are the prospects here? I mean, the idea that we have these already approved drugs that we know how to use is appealing, but it's also often very hard to take a new drug and make it work in a different disease.
You know, I agree with you. I think the likelihood for success is much higher with drugs and vaccines that are tailored to make a difference for COVID-19. The thing is, of course, even if you work very fast, it takes, you know, we still are having another 6+ months before we could have at least 1 million doses of vaccine, touch wood, if things go well. But it may be, you know, another year until you have billions. And the same for antivirals. So, you know, you need also to be curious in the sense when investigators put existing learnings about this disease into a proposal to test something that's already present. You just need to be very realistic that most of them would fail. And we need to understand the science of this 4-month-old disease better. And, you know, I do think that you hear a mixed readout on the IL-6 inhibitors that you mentioned, Regeneron and Roche. But, you know, it may not be that all patients are the same. I think we have learned that some patients get early on more what you call innate inflammatory reaction. And what does that mean? It's the old ancient part of the immune system, not the adaptive that develop memory, that seems to be more in this IL-6 cascade. So I think that we should still, you know, learn more about the science and ask ourselves whether some patients may benefit, but maybe not all. And, you know, Xeljanz is not approved for COVID-19. So when used by investigators, it's an experimental setting, but its profile is more of a modulator. It does not only act on IL-6 like these antibodies, it does modulate and reduce IL-6, but it acts also on a lot of the T-cell cytokines that we think are part of the transition from a more initial burst to a sustained burst of the immune response. So I think it's a very reasonable hypothesis to test under experimental conditions in agreement with patients and physician dialogue, some of this new treatment, and at the same time try to maximize the learning. In addition to Xeljanz, we also have plans to try another drug in this series of signal transduction inhibitor that's called IRAK4, which we think are particularly active on this more ancient macrophage-like immune system in the initial burst, but it goes a little bit broader than IL-6. So we try to contribute with investigators when they approach us to cast a wider net and hope somewhere in that wider net will catch one of the ugly fishes that make the disease worsen here.
Yeah. So a really good question from Kristen Schneeman here. Are you concerned about having enough patients for well-powered studies as time passes and the number of new cases we hope falls? And there's so many products being tested in studies and entering studies. Like, are we going to end up with if we're hopefully on the downside of the peak of cases, are we going to have enough patients to test all these drugs?
Yeah, I think this is maybe a very increasingly logistic problem. I think there is still going to be for quite some time a pandemic, but it may move around. So when you plan a clinical trial, you probably need to have a very broad footprint of investigators, whether we speak about United States or different continents, so that you're able to use those who are at the very moment of your study that starts to be a peak and people suffer. The other piece is that we're putting a lot of work in addition to try to generate what you call event data, Matt. So if you have a vaccine, you immunize 1,000 people, and maybe if there is 5% in the population that would get the disease at a given time point, 50 would get the disease. And you hope your vaccine may bring it down to 5. But of course, to bring that to a scale where you feel like absolutely comfortable with data, particularly if the incidence of the disease is moving fast and it's hard to follow different regions of the world, what other biomarkers of likely vaccine efficacy can we identify? So we are performing, in dialogues with the FDA, studies on primates and likely a smaller animal, maybe hamsters, where we will immunize them with a vaccine, expose them to SARS-CoV-2, and then see at what level of vaccine-induced immune response are they protected. Mm-hmm. Now you could argue, well, that's really helpful, but it's antibodies produced in animals. Mm-hmm. So I think another clue to this Sherlock Holmes work would be to take insights from what's called convalescent plasma treatment. Mm-hmm. And you and others may have seen that many hospitals, before they have a new generation of therapeutics, are asking patients that have well survived the disease to donate plasma. And they want to purify the antibodies and show that they react with SARS-CoV-2 and give them to patients that may be more vulnerable. So I think from those studies, you may also be able to learn what is the level of antibodies that seems to give an impact on humans. Can you even use some of those antibodies in primates and protect them? So this is the puzzle that we think by August, September, working as an ecosystem of companies, academics, regulators, that may help us to say, if you have this type of antibody response using well-characterized assays, you are very likely to be protected from infection. Mm-hmm. So you can add that to event data. And I think it could help us to get confidence to use the vaccine earlier, assuming safety.
Mm-hmm. I mean, how much this varies by vaccine? How much does an antibody, I mean, there are cases where we don't develop antibodies naturally, where we've figured out how to make the body create antibodies through a vaccine, correct? So, I mean, that's certainly the case with any experimental HIV vaccine. That's the whole challenge there. How much will you be able to compare between antibodies created through convalescent plasma and the vaccine antibodies? Because they're going to be different, aren't they? Aren't they functionally different antibodies?
You really pinpoint what I think is important here, rigorous science. And it's a little bit what makes me nervous when you see all these diagnostic tests that actually are aiming to measure a lot of antibodies. Unless you're really rigorous, and some may, but it's not always specified. You need to really measure what type of antibodies do you have. How do they bind to the virus? And are they neutralizing? When you have those zip codes, you can then compare with antibodies in your vaccine. If you don't have that zip code, where do you bind? What type of antibody are you? Are you neutralizing? And how potent you are? It's basically a 4-letter zip code. And if you don't do that rigorous work, the envelope may end up at any place. It's like you don't write the correct email destination and it never reaches the recipient. But even with that rigorous, you need to think about, are antibody the only way to protect from this disease? We do think they are a major component, but likely also some of the T cells, the adaptive part of the immunity, are important. And we will be following them in those type of cells also in our clinical trials. We think the antibodies are the most straightforward, but we think the cellular response may help us to feel good about sustaining long protection and low risk of unexpected negative immune reactions.
So we're coming to a close here, but I wanted to give you a chance for closing remarks, but I also wanted to just confirm on the timeline here. You guys there was some discussion in the analyst notes yesterday. You actually are imagining that an EUA or an accelerated approval might be possible this year for this vaccine, that's a plausible timeline.
Absolutely. Like we discussed, Matt, you have to put a moonshot type of goal in front of you. This program is called Lightspeed program. It still means you do science rigorous, that you are extremely attentive on safety. We believe, pending good data, and that we can continue to work this closely with regulators, that it's absolutely feasible to have a vaccine emergency use or accelerated approval in October. And that's what we're aiming for, because then we will start to see flu. And I'll hate to see that we are, not well prepared to be hit by two very difficult, one epidemic and another pandemic. If you think that it was hard work at the hospitals, and I admire and want to acknowledge, I had an opportunity, since I live in New York, to get to know a lot of the New York hospitals and people at those departments. It would be a nightmare scenario if we have two viruses hitting us at the same time. So that's really our goal. But as I said, you can't protect all of the continents at the very same time. You start with the most fragile, you start where you have the most epicenters of epidemics, and then you move forward. But it's only possible if we work as a community together. And I was, of course, inspired when our CEO, Albert Pola, approached us and even publicly and said, we need to work completely differently here. It's not about, looking at spend to get to this place. It's our mission to be part of those providing a solution. And he launched a 5-point plan about transparency, working with others, helping out, sharing data. And I've been so, thrilled to see everyone in the pharma community, biotechs, medical centers, regulators, NIH, respond so quickly. When our antiviral program started to burst and we wanted to accelerate it, we didn't have all the assays in-house. And actually, I sent an email to Francis Collins in a late evening and asked Francis, could you share with me which labs in the United States may be the right one? Because nobody had published any data yet. Half an hour later, he connected me to someone at NID that has been just brilliant in the engagement to create that network. And the same has happened with so many people, whether scientists or physicians or those representing patients and nurses. So I think we have stepped up as a group together. And if you wanted to say a closing remark, I think we need to work very focused on looking upon this as a multi-pillar approach. I welcome, those initial attempts to use repurposing of drugs, and we spoke about some of them today, that were used in other areas, and explore if they immediately can be available, including convalescent serum. The longer-term solution is highly potent therapeutics that can be used, infusions for those that are in hospitals, and it can be used for patients that are sent home to be earlier not contagious and have limited to no risk to end up later in hospitals. And then we need absolute solution of vaccine or vaccines that can protect the population and expel the virus as much as we can. We need to be diligent and continue to survey for the virus. It will try to come back. It will change shape. We will see siblings and cousins of the coronavirus try to jump again. So we need to keep that arsenal of tech platform to be able to create a more peaceful environment with this type of viruses. And that journey we're on as scientists. And I have no doubt that science will win, but it's going to be a long haul. But I feel very good about how we will be able to change this devastating few months we have suffered together. and we can only do it as one committed community. I want to applaud you invited me and Rich to be part of this dialogue with you, and all the great questions that we had today. They will continue to inspire us to think about anticipated events in this journey, to have something ready as soon as possible. Feel welcome, Matt and others, to stay close to us, share new ideas, and let's stay optimistic, but at the same time base our predictions on proven science that repeatedly is seen by laboratories. And we have to be cautious in early clinical trials that are not well controlled. And I think we learned it from this period, but it's not a sign of complete failure. It's a sign of people doing their best under extraordinary difficult conditions. And I think it all plays into learning of the community of scientists, patients, physicians, and everyone.
All right. And with that, I think we're a little over time, and so we'll wrap things up. Thanks again to our event sponsor, Pharma. Thanks, Michael, for joining us. Thanks to all of you out there for attending and asking such great questions. I'm sorry I couldn't get to all of them. A reminder that we will be hosting our third and final installment for this Combating COVID-19 series on Wednesday, May 6th at 1 p.m. I'll be joined then by Moderna's Chief Medical Officer, Tal Zaks, to talk all things COVID-19 vaccines, or really about mRNA vaccines again. And I hope you'll join us. Thanks again, all. Stay safe, really stay safe, and be well.
Thank you so much.
Thank you.